Hi, I'd like to thank you. Hopefully you guys will stay awake before the next break. And I want to thank the Every Life Foundation for the opportunity to present a little bit about our company and a case study on our lead clinical program. I'm with a company called Fibrocell. We are a cell and gene therapy company, and if we're talking about stats here, we're a very lean company. We're a public company. We are 20 people right now, so we fall into that category of trying to do a lot with very little resources. As a public company, I need to include this forward-looking statement slide. And so today, the overview of my talk will be, I'll provide a brief overview of the company. Our lead indication is for an indication called recessive dystrophic epidermolysis bullosa. And I want to specifically talk about one particular challenge that we've been facing, which is in wound healing, which is one of the key presentations of this disease. And then I want to talk about the recent initiatives that have helped define the path to move our therapy and other therapies forward for this debilitating disease. And then finally, I'll end with some considerations for thinking about a rare disease center of excellence. So the history of Fibrocell is that we've actually were a company called Isologen that was formed in 1995. And they developed a product called the autologous cellular systems, which is basically taking a biopsy and expanding fibroblasts and using it for cosmetic applications. So this product was actually used in about 1,000 patients when the agency said this is a product that is actually more than minimally manipulated, so it actually requires FDA approval. So at that time, the company established a GMP facility, SOPs, and all of the other things for approval for this product. So after 2 pivotal phase 3 studies under SPA, which met all of the primary endpoints for nasolabial folds, this company basically got the advisory committee for approval. It recommended it for approval in 2009, and then the BLA was approved in 2011 for a product called Lviv. And at that time, during that time, there was a reorganization of the company and rebranded itself as Fibrocell. And Lviv was the third autologous cell biologic product approved by the FDA after Carticel and Prevenge. And subsequent to that, Fibrocell collaborated with a synthetic biology company called Intrexon for the next generation autologous fibroblast platform where they genetically modify the cells. So from that collaboration, the first product that we're developing is FCX007, which is a cell and gene therapy for RDEB. So again, this is a personalized biologics approach where the delivery vehicle is the autologous fibroblast. It's readily sourced. The fact that it's autologous, you have reduced rejection and immunogenicity concerns. And based on the Lviv pathway, we have an established manufacturing process and regulatory pathway. We use the autologous fibroblasts where we take the biopsy, we expand them. We then use a 3rd generation lentiviral vector to transduce the cells ex vivo, and then we basically expand the cells further and then provide the therapy via local administration. So this is our developmental pipeline. We have 2 programs in the clinic. The first one, which I will be discussing further for RDEB, it's in phase 2 right now. We have orphan, rare pediatric, and fast track designations. And our second program, FCX013, is for moderate to severe localized scleroderma, where the target here is MMP1 using an inducible promoter to basically reduce fibrosis in this disease. So today, I'm going to speak specifically about recessive dystrophic epidermolysis bullosa. And this is one of several genetic skin blistering diseases. And this one basically is, in this cartoon, I'm depicting the skin. So at the very top is the basal keratinocytes of the epithelia. And then you have the basement membrane zone. And then below, you have the dermis. And in the cartoon on the left, it's showing in green what are called anchoring fibrils. And this is a protein structure that is encoded by the Col7A1 gene, which encodes for type 7 collagen, which is the primary component of these anchoring fibrils. And so you see that in normal skin, it keeps the epidermis and the dermis together. In our dead blistering skin, you can see that due to mutations in the gene, you have either reduced or absent anchoring fibrils, and that allows for just very minor friction to cause blistering. And this you can see on the panel here. So these patients basically have blisters all over their bodies. It's very severe. They're typically continually bandaged and to prevent further blistering. So it's very devastating. It's very painful, and this is diagnosed at infancy. And because of this constant blistering, there's a very high mortality rate, and most patients do not live to their 30s, and typically death is due to squamous cell carcinoma, infection, or malnutrition. So there are no approved therapies for RDEB, and the current treatments only address the symptoms. And right now, this basically encompasses bandaging and antibiotics. These alone can cost $10,000 a month. And that doesn't exclude all of the other interdisciplinary care that these patients have, including, so this doesn't only affect the skin, it also affects epithelialized surfaces, including like the esophagus. So they have what are called strictures in the esophagus, so they have to have surgeries to remove those, as well as what are called mitten deformities. And a lot of that is like you have contractures, and then they have to have hand surgeries and other things to kind of remove that so that they can regain function. So again, the clinical manifestations, as I mentioned, is this unremitting skin blistering that evolves into chronic wounds, inflammation, and fibrosis, and this cycle continues. And here's an example of a patient with the mitten deformity. And the causes are, we know the underlying defect for this disease, but in terms of why they have very aggressive squamous cell carcinomas, there's some recent research that is kind of highlighting, where that's going and why that is. So I want to talk about the clinical challenges of trying to develop an outcome based on wound healing. So there was a long study, the National Epidermolysis Bullosa registry that basically looked at the long-term sequelae. So they had a lot of the other problems that were documented, like anemia and other things like the squamous cell carcinoma and other things. However, there was very limited data about the natural history of the wounds themselves. And this is very important because these wounds are very dynamic, and that just hadn't been very well characterized before. So this was recently addressed by a large cross-sectional study. And basically, this study showed that these wounds could be categorized into two buckets, if you will. So you have wounds that are chronic, it's defined as areas that are unhealed for over 12 weeks. And then you have areas that heal, but they reblister fairly quickly. So those are called recurrent wounds. And so once we had this definition of what a chronic wound versus a recurrent wound, that actually will help in terms of defining clinically meaningful efficacy endpoints. And I think the other thing that this study had showed was basically that these wounds are significantly different from other chronic wounds, such as diabetic ulcer wounds. And the significance of that is that we were looking to the guidance for chronic cutaneous ulcer and burn wounds. And in that guidance, it basically says the complete closure of a chronic non-healing wound is one of the most objective and clinically meaningful endpoints. However, this natural history study of these wounds showed that these wounds are dynamic and very different from other types of chronic wounds. And more recently, there was a study by another group that is also using a gene therapy approach for this disease, where they have transduced keratinocytes, made them into grafts, and then using these grafts to treat the RDEB wounds. And when this study was done and taken into consideration of patient-reported outcomes of pain, itch, and ease of blistering, it was shown that wound healing of 50% actually had statistically significant improvements in these PROs, which is pretty important considering 100%, if that's what we're just thinking of as being clinically meaningful, but for patients, that are living with this daily and realizing that, anything to help with the pain and itch and the blistering is going to be helpful for them. And this leads into all of the events that have happened recently. There has been this externally led patient-focused drug development meeting with the FDA in April of this year. And this was basically a meeting convened by Deborah of America, and it allowed patients with EB to provide testimony of their symptoms, the issues that these patients face, and the importance of therapy and what it means to patients. And what was surprising to me and very responsive of the FDA is that within 2 months that they were able to provide some draft guidance. So the epidermolysis bullosa developing drugs for treatment of cutaneous manifestations guidance was provided in June of this past year. And I think what's really important is how it acknowledged that the distinct pathophysiology, natural history, and low prevalence of EB warrants distinct approaches to this. And I think that was basically their nod to say that, yes, this is not exactly like other chronic wounds, and different approaches need to be taken. And I think having understood the unmet need and the natural history of this disease, it basically has informed greatly our target product profile and our clinical study designs, and suggests that maybe a path of less than 100% wound healing may be an important clinical endpoint here. So I'll present a little bit about our phase 1/2 clinical trial design. So this is basically to evaluate a single intradermal injection series of FCX007. And our secondary objectives are basically to use both immunofluorescence to see the type 7 collagen that forms from these fibroblasts going to the basement membrane zone and actually creating the anchoring fibrils. And we're also looking at wound healing. So this is an open label study where, again, if I go back to talking about why it was critical to understand the wounds here, we basically have to monitor, we do this wound monitoring phase before we actually dose the patients to understand which type of wound there is, because you can imagine if you are treating a chronic wound, but then you have as your control match size a recurrent wound of the same size, you could see how that, if that recurrent wound is basically healing and reblistering and healing and reblistering while you're measuring over time, that could confound your results. So it was very important to understand that. So we use a matched untreated wound, as I'm saying, for in the same patient as a control. And this is just showing our sites at Stanford University and Children's Hospital in Colorado. Our phase 1 was completed with 4 patients enrolled, and we're continuing to follow up those subjects for 52 weeks. And we have now dosed our first patient in phase 2. And the transition from phase 1 to phase 2 was also that we submitted data from the adults to the agency so that we could enroll pediatric patients. And now we are enrolling pediatric patients age 7 and older, and so far have enrolled 3 pediatric and 1 adult patient for the phase 2 portion of the study. So I'm going to show just some brief data that we presented at the International Investigative Dermatology meeting this past May. And this was based on the original 4 patients that were dosed. The age range was 20 to 37 years of age, and we treated 7 wounds ranging in size from 4.4 to 23.2 centimeters squared. And the cell dose concentration was 14 to 25 million cells. So we inject these at the periphery of the wound, as well as the axis of the wounds, and basically look to see over time if we see an increase in collagen 7, as well as what happens to wound healing. And I'm not going to present all of the data because of time. I'm just going to go ahead and show that wounds that were treated with FCX07, if you look here on the middle column here, it's basically showing that 50%, greater than 50% healed compared to the baseline. And at 4 weeks, we showed that all 7 wounds were basically healed. And then basically, if you go out 12 and further, we still see pretty good wound healing effects. And what is important to note here is that previous studies using allogeneic cells showed that while you show improvement in wound healing for the first 28 days, so for the first 4 weeks, subsequent time periods, it just deteriorates and the wounds re-blister. And here at the bottom, I'm showing the untreated control wounds where just 14% of the wounds basically showed greater than 50% healing compared to baseline at 4 weeks, and did not improve over time. So the goal of presenting this today was to just highlight how all of these events have been very instrumental for moving our program forward this year, including how the EB guidance has helped us, and also what Dr. Bryan talked about this morning about the gene therapy for rare disease guidance. In assisting us for developing therapies for rare diseases. While this is wonderful for our program for EB, how does this apply for other rare diseases? And I think that's the question that we're all trying to understand here, because not every disease has guidance. For example, we would love to have guidance for our localized scleroderma program. But I do like this idea of having a rare disease center of excellence, not necessarily for the specific medical or clinical applications, because that's going to come from most of these rare diseases. There's only going to be a handful of people working on each indication. But I do think that as Dr. Kakis said, the methodologies and other things can really, really help just the sponsors understand. So we're not guessing, like it's not, we're just trying to shoot from the hip to try to figure out what is the best path here. And I think if we can have some sort of clinical review across all the divisions and have one group that really focuses on like the statistical methods and other things that are really important for rare diseases, I think that could be really helpful and appropriate for sponsors working on these indications. And the other thing that I thought was very important, and Dr. Bryan mentioned this earlier, was this idea about starting natural history studies early. And I think this is also where a center of, rare disease center of excellence could help facilitate a single, well-executed, so you're not just pulling, random bits and pieces from other places and maybe make that more effective, right, for all of these different indications. And then finally, I don't know if, how helpful this would be, because there probably aren't many therapies being developed for all of the same rare diseases. However, for example, in EB, it is fortunate that there are a number of companies developing therapies for the different subtypes of EB. And taking a cue from the oncology space, I wonder if like a master protocol, where you have one protocol, and although it is going to be very difficult, but you have multiple sponsors, lots of stakeholders, multiple therapies, but because this is a small patient population, is there a way to maximize and get the most beneficial data, right? And it seems to me that the FDA is in the prime position, because as some people have pointed out, they're able to see so much more data, and it would be a neutral party, right, that could kind of administer something like this, might be something to consider. So with that, I will say thank you very much and answer any questions.